transcription in prokaryotes, and I am discussing about the process of termination in this particular video. So termination, what happens during termination is there are two type of events which can lead to the termination. One we call is Rho-dependent and another is known as Rho-independent. What is Rho? Rho is kind of enzyme. Rho is kind of an enzyme helicase which breaks the RNA and DNA hybrid and then releases the RNA. And then why is these called extrinsic whereas these called intrinsic? Because the termination in this process is brought about by external factor, right? And the termination in this process is brought about by the mRNA itself. Alright, it is brought about by the internal factor and this is brought about by external factor, that is with the help of enzyme, the enzyme or a protein known as Rho, and this happens with the help of the mRNA which has been made. That is why it is known as intrinsic and this is known as extrinsic. Now coming to what happens in the Rho-dependent process. In Rho-dependent process, what happens is the helicase-like enzyme, which is a 6-hexameric unit protein, it get inserted in the mRNA and it goes in a rotating manner. Alright. So what happens when the stop codon UUU comes? Then the RNA polymerase, after addition of UUU, RNA polymerase stop for a second. When the stop codon comes, then what happens is RNA polymerase has ability to trace back. Alright, RNA polymerase has ability to trace back to check if any wrong nucleotide has been inserted. So when the conjugated UUU comes, then it waits for a second for what is happening if I am doing something wrong. At that time, what happens is during this time, Rho catch up and break the bond. At this particular time, what happens is Rho will catch this thing. It will wound and rotate and it will enter and then it will just break the bond because RNA poly has stopped for a while. Alright, in this what happens? The external factor, that is 6-hexameric unit protein which has a helicase activity to break the DNA and RNA hybrid, enters the mRNA strand and then it rotates. It goes inside by the rotating fashion, and when the stop codon reaches, the RNA polymerase will wait for a second. At that particular time, Rho will catch the hybrid portion and then break the hydrogen bond between them, and this mRNA will fall off. Alright, now coming to Rho-independent or intrinsic process. In this process, what happens is this is my newly formed mRNA, and what happens is after some point this UUU is added. Alright, and we know that A and U is a weak force. They are only bonded by 2 hydrogen bond, whereas G and C is a strong force, right? What happens is after some period of time, after the new mRNA is made, after the mRNA is being formed, there will be a palindromic sequence known as palindromic sequence. Palindromic sequence is a sequence, if we read it from the 5' to 3' and the 3' to 5' and it will be same. You see this sequence from 5' to 3' and 3' to 5'. You match it, 5' to 3' and 3' to 5', it is same. Alright, and then what happens is that there will be a repeat. See, in this is mRNA strand which is made. Alright, within that mRNA strand, what happens is we will find some kind of inverted repeats. Inverted repeats means like this is the mRNA strand, and within this strand itself there is a complementary sequence. For example, this is the mRNA sequence and this part is complementary to this part, then what will be happening? If see in this particular sequence, for example, this is the mRNA sequence, and if this part, right, is complementary to this part, then what will they do? They will try to bond with one another, right? They will try to make a bond with one another. See, GG and GG. Now what they will do? They will make a hair pin like structure and they will bond with one another as this part is complementary to this part, right? At certain point in an mRNA, in this process what happens? At you just note it, at certain point in mRNA there will be an inverted repeat of GC. All right, there will be inverted GC repeats, and these repeat being complementary to one another form a hairpin like structure, and this hairpin like structure is, they form a RNA hybrid. This hairpin like structure, it is forming RNA hybrid, one thing, and this is DNA and RNA hybrid. Now it is said that always RNA hybrid is always favored as compared to DNA RNA hybrid. The reason of breaking of these mRNA from the template DNA is because RNA hybrid is always favored as compared to DNA RNA hybrid, and that is why this is much stronger as compared to the DNA RNA hybrid. And another thing is, this RNA hybrid is bonded by GC base pair, and we know that G and C, they bind with one another by 3 hydrogen bond, and it is much stronger as compared to A and T, which is bonded to one another by 2 hydrogen bond, right? So many things favors the falling of this mRNA from this particular hybrid. So now what happens is, this particular thing will fall off, right? This mRNA will fall off, and then there will be the synthesis of protein from this mRNA. You got it or not? Okay, now I'm just going to recap the thing. There are two processes of termination. One is Rho-dependent, which is known as extrinsic because it is brought about by some external factor, and it is the Rho-independent is known as intrinsic because it is brought about by internal factor. In Rho-dependent, what happens is, there is an helicase-like enzyme which is made of 6 hexameric unit of protein. It will wound inside the RNA, recently formed RNA, and it will reach the DNA RNA hybrid, and the point in which there will be the addition of UUU, RNA pol will stop for a while, and at that particular span of time, the Rho will catch the hybrid portion and it will break the bond. In this, what happens is, there will be a formation of hairpin like structure. Because there will be GC inverted, there will be the formation of GC inverted repeat in the mRNA sequence, and then the GC will bind with one another forming RNA hybrid, and due to the formation of this GC RNA hybrid, there will be the strong forces in the strong and weak interaction between strong and weak forces. Due to the interaction between strong and weak forces, what happens? Strong force wins, and finally mRNA will fall off from the template DNA. Okay, this was only for today. Thank you.